Okay, good morning. Welcome everyone. This is lecture 11, and we'll be focusing on the interaction between human and microbes, right? And more specifically, we'll focus on two areas: the microbes on the surface and inside our body, and then how we can control their growth. So microbes, we know microbes are everywhere. And then there are two main types. We can group them into normal flora, that those are the microbes are normally there. So on every inch of your surface, your skin, there are many microbes there, right? But then there are harmful bacteria normally are not on your skin, or actually on your skin, but to the wrong place. Okay, so for example, the E. coli is normal in your intestinal tract, but if you somehow got the E. coli in your eye, that will cause an infection, right? So that's the type of the distinctions that we're drawing from normal flora and harmful bacteria. And then in terms of control, how do we control? There is two main types. One is the general host defense mechanism. One is a specific. It is important that you understand the general. Here's the concept. General is that it applies. It's a mechanism. Those are the mechanisms that apply to every microbe. Okay, and specific, in contrast, then is to apply to specific bacteria or microbe, which means for the specific host defense mechanism to work, you will have to identify which microbes you are trying to control. All right. So we're going to start off by, and so these two are the microbes and us. These are really interacting. And so in this lecture, what we'll do is that we'll first talk about microbes and what are the things they do to overcome our defense mechanism. And then we'll talk about our defense mechanism, how we trying to keep the microbes out. And then in the end, I'll have a few slides to discuss how these things are forms a network of interaction, because they do. All right. So when we think about normal flora, and those are normally on the skin, oral, and gastrointestinal tract. And so we want to talk about the balance. And so we first talk to the skin. And the skin, as I said last lecture, is actually a very tough protective layer for us. You have the epidermis, dermis, and so the skin on the surface is very dense. It's no bacteria can really penetrate. Okay. Now the weakness on the skin for infection is these hair follicle glands. If you have pimples when you're growing up, and that's where the microbe can go in. And it's really, once they're there, it's really hard to get rid of them. All right. And so these are the because we have glands, and then you produce sweat, and so liquid and nutrient. And remember, our waste product is always someone as nutrient, right? And so they're there. However, the most important message we want to get across is your skin is really very good at protecting you. So as long as you don't have physical cut, having a few bacteria on your skin, it's okay, and they're dry, And for most part, they're dry because they're exposed to the air. They're tough. And so that's how we can survive. Remember, we survived in the woods, a few thousand years ago, right? And then one student is reporting having problems with audio. Anyone else?
Like some static is coming across.
Okay. Not sure. Because I have my usual setup. Okay. Send me a reminder if this keeps keeps occurring. And so so aside from the skin, the cavity, the digestive tract actually is another major area that we expose to microbes. Actually, have majority of microbes are in the digestive tract. Well, it starts with the mouth, right? The oral, and so there are lots of areas microbe can grow, okay, like this area, and they keep growing and growing. When you are young and energetic like you yourself, your bones, teeth bones, are very strong and hold them together. And when you get to my age, and the bones start deteriorating, I wish there's mechanism for growing these bone back, but they have lots of we have microbes. And then go down the digestive tract, and this is a rough sectioning of the digestive tract. And so you have stomach, small intestinal tube, and the large intestinal And yes, go ahead. You have a question?
Yeah, when you were talking about the oral cavity for when you get older, so when you get older and it starts to like deteriorate, is it easier for microbes to get in? Or were you just saying that just to say it?
Yes, it's always easier yeah, because when you are older, the microbes are here, which I'll talk a little bit. But when you're older, the top part of the bone is gone, and it's easier for the microbes to go in. Okay, so they can continuously go down, and when they get here, then eventually these roots are rotted, and then you lose your tooth. So what I like to highlight is that these are on this side. Okay, so on these, this is the major microbes, the bacteria on different parts of your digestive tract. Okay, you can look for E. coli is here. And what's amazing is this part, the small intestinal tract. It's very, very long. Okay, and but it has just really these two, Enterococcus and Lactobacillus. And in part, or mainly because of the low pH, right? The pH is very acidic. You see it's here, 4.5. Well, stomach is the lowest, right? Normally, bacteria doesn't grow, but there are some bacteria can colonize your stomach, and like this Helicobacter pylori. And this is one bacteria that the doctor will plays a complex role. When you're younger than 40, like you are, this bacteria actually helps protects you. Yes, go ahead with your question. Did you have a question? No? Okay.
I forgot to take it down, sorry.
Oh, okay, that's all right. Um, and the pH, and then stomach has low pH, very low pH, but some bacteria like Helicobacter were able to grow because they are initially, especially from your mother, when you're a baby, the Helicobacter of your mother typically gets to the fold of the stomach because when you're a baby, your stomach isn't that acidic, and then the bacteria colonize it, and then they actually provides a little some protection, innate protection, and then but when you get to 40 and 50, and that bacteria tend to be associated with stomach cancer or an ulcer, and that's when your doctor is going to recommend you get rid of it with high dose of antibiotics. And my doctor treated mine, and we haven't gone back and examined whether I still have Helicobacter from my parents. Okay. Ania, did you have a question?
Yeah, so you only get this from your mother?
No, you do get it from everywhere. As you grow older, these bacteria can colonize, but what's reported is that the ones from your mother actually persists a lot more than the one that you get as a teenager or 20 something or 30 something. All right, and so if you like, New York has its own Helicobacter uh, flora, and but if you go to LA, you're going to get some other Helicobacter. But then because you already have one colonizing the from your mother, and then that actually prevents the other Helicobacter pylori from infecting. And that's part of the innate defense mechanism, which we'll talk about. Yes, Catherine?
So it depends on the diet of the person, since the diet is like kind of different in each region. That contributes to
No, no,
like
no, no.
how
It's
the
not the diet. Well, maybe it's related to diet, but but nowadays we keep all the food clean, right? And so, but it's never going to be sterile. So yes, there will be some bacteria will get into your system, digestive system. And then the other important part is the respiratory tract, and which you probably become much more familiar because of the COVID. It can be grouped in separated into the upper and the lower. And what's important that you understand is that the respiratory tract has no, especially the lower, has no real physical barrier, right? So because of the need for air exchange, right? So what we do have is we use mucus, and so the mucus is coating all the respiratory tract, and as the air goes down, the particles or the virus or bacteria particles in the bounces around, and the hope is that will hit the mucus and then will get trapped. And so that's why the concentration of microbe in the air is important. And if it's a low concentration, then like fresh air, and then when the most likely, almost certain, all the microbes gets hit and trapped in the mucus. However, if someone next to you just coughed or sneezed, then you have a high density of microbes, and then some microbe can escape avoid being trapped by the mucus, and that could cause an infection. So those are the areas. Not so the barrier. What I yes, the barrier. So I was comparing the respiratory tract surface versus the skin. Skin has a tough layer, and we don't have mucus, right? We rely on dense skin to protect us. The respiratory tract relies on the mucus to protect us. That's the difference. And so knowing this, then the microbe, well, remember, microbes are only interested in getting food. And where are the food? Well, food is inside our cell, right? And so microbes will colonize and break down. So they will get across the mucous membrane and then grow and then break down the cell and then cause an infection. That's a general problem or general route of infection. So then if you think about stomach, and in stomach, normally the pH will be very low, but the Helicobacter pylori, when they are colonizing the surface, they will produce ammonia to neutralize local pH. So they, especially if you think about stomach is not flat, has lots of folds, right? Because it's stretch and shrink. And so it's in those folds that the microbe can colonize and then change local pH so that they are not exposed to pH 2. They're more like pH 5, right? So they can grow. And if you have harmful bacteria trapped, and then they can break down the mucus, break down the epithelial cell, and cause an infection. Now back, Ania, yes?
So in part C, that's what that's showing, the part where the microbes are colonizing and causing infection.
Yeah, and so this is where the infection is, right? Whenever you have microbes getting into tissue, culture, cell, and that's an infection. So if we think about the microbes in the mouth, and that's where the first part of our body that contacts or open to microbes, colonization. And so this is the dental plaque. And so on day one, and if you stain for the dental plaque, which is a mixture of bacteria and food particles, and the microbes tend to stick them together. And if you wait for 10 days without brushing and you stain it again, and this is what you actually will see. Now, under the microscope, that will be different. That's what, when you look into the microscope, this is what you see. And initially, you have microbes individually, but when you let them grow longer, and this is what you'll have. And Streptococcus mutans is the dominant microbe in your mouth. Okay, that's a dominant microbe in your mouth. And what you can barely see is the long string of individual bacteria, but they are covered in a layer of mucus. And they produce this mucus in the presence of sucrose. And do you know where you find sucrose? What is sucrose? Sucrose is a sugar from... Yes, go ahead, Mamadou.
It is glucose and fructose that gives sucrose.
Okay, that's a very good, right, Mamadou. But where do you get sucrose? Ah, yes, Lucero, from sugarcane, yes. And it's the most well, it's the cheapest sugar that we have. And so actually it's everywhere. The candies and added sugars. And what sucrose will happen when Streptococcus mutant, this bacteria, runs into sucrose, the two meets, it will do two things. It will produce this polysaccharide, right? You see this polysaccharide layer of like a glue-like thing, and then they stick. You can even see the model. They stick themselves like a glue. They glue themselves to the surface of the mouth, and then they ferment. When they ferment, they produce acid. Okay, so when you have a bacteria stick to your tooth and produces an acid, it's almost the same as a dentist put a drill in your tooth and start drilling, and constantly. So that is the reason why babies, especially babies, tend to lose that, have tooth decays, holes in them. Okay, so so if you obviously you want to reduce the number of sugars that you take, but what's best is if you can't stop having candy, but make sure you have candy, but don't go to sleep with candy, right? Especially, and then after you have candy, you make sure you wash your mouth and remove the residual candy. Okay, so that will help. And so having the baby with bottle and go to sleep, and that's just the worst thing that could happen for their teeth. And so soon you are all gonna have you will have babies, and hope you'll remember that I told you no bottle. Baby shouldn't go to sleep with bottles. And then you can tell them this is the reason. The tooth, they will lose all their tooth because of the sucrose. And it's interesting that glucose itself doesn't do as much damage. It's the sucrose. Okay, if you want to know more, we can spend more time talking about this. But now I wanted to go back. So those are the normal bacteria. Now I wanted to go talk about harmful bacteria, because that's very important. As you can imagine, for harmful bacteria to infect you or us, the infection would go to steps, right? So they will go through these at least 4 different steps: attach or adherence, invade, meaning break over the barrier, and they colonize, meaning they grow, and when they grow to larger extent, they will produce toxin. And so this is a graphic representation. So the bacteria is on the surface, skin, or mucus, more like a mucus. And then they will break through the layer, and then they colonize. And when they start colonizing, and you have lots of bacteria growing, and lots of bacteria, they produce lots of acid, lots of enzyme to break down. And so they produce toxin, and then they produce toxin that can be circulated. So the local, right? Local, they have enzyme to break it down, and then they can have toxin circulating through your body. And their goal obviously is to kill us, right? And so they can cause tissue damage locally and remotely too. Okay. So that's a general process. Now, the different bacteria have different levels of infectious, how bad they are, how infectious they are, right? And how virulent they are. And so so we have a measurement to measure how deadly the bacteria that is for any given bacteria. So this is the measurement is LD, lethal dose 50. So LD 50. So here is a graphic representation of LD 50. So on the y-axis is the percentage of mice killed if you give bacteria to mice. And then you can inject different numbers of bacteria per mouse, right? So you go from 10 to 1 million to 10 million here. So if you look at this, if you inject, say, 50 Streptococcus pneumoniae into a mouse, the chances are, and if you do that for 10 different mouse, and 50% of them will get killed. And if you inject 100 bacteria cells, Streptococcus, you kill almost 100%. However, if you inject the same number of bacteria into a Salmonella into mouse, see only a small percentage of bacteria mouse gets killed. You would have to inject 500 or 5,000 Salmonella into a mouse, into one mouse in order to kill, versus you inject 50. So it's the 50 versus 5,000. Okay, so compare these two, your conclusion of which bacteria is more infectious.
Streptococcus.
Yeah, so it's clearly Streptococcus, right? It's 100-fold more infectious. So that's the LD50, and then we need to think about how much toxin is produced and how invasive a bacteria is, right? And so these two concepts are, one is toxins produced, chemicals, and invasiveness is referring to how bacteria can avoid being detected by our immune system and then continue to spread throughout the body. And in some way, these two features are linked. One helps the other. But mostly it relies on they have to produce what's called the virulence factor. And these are the chemical molecules that are produced by microbes for various purposes, okay, mainly to avoid, to defeat the host defense mechanism. Okay, so here's a graphic representation of some of the features. O antigen. Okay, so remember last time we talked about a bacteria E. coli called O157H7, and the O referring to the O antigen. It's the polysaccharide on the surface. And it helps the bacteria evade human defense mechanism, which is the phagocytic killing. Okay, so O antigen, if the bacteria produces an O antigen, our body will have harder time to kill them. Okay, and then you can have the flagella produces function as the antigen that helps the bacteria attach and bacteria avoid the phagocytes. Yes, Estefani, sorry.
Hello, Professor. The O antigen
Yes, go ahead.
has to do with like the RH or the ABO, right? It has something to do with that. It won't affect the
Say it
body.
again. I'm sorry. I say it again.
I was saying that the O antigen, it doesn't affect the body, right? So if I'm imagining like ABO, like blood type, the body won't have like a weird reaction to that virus, right?
No, those are two different things. Blood type are the surface molecule on your blood, and O antigen referring to very specific to the polysaccharide on the surface of bacteria. It's different. So the O antigen is not like the O type of blood. Yeah, and then you can have toxins like enterotoxins, and these toxins actually we'll talk just a little bit later. And these toxins can be produced by bacteria and then cause diarrhea. All right. Adherence is just attach. Adhere is the word that attaches. Like the adhesive, right? You know the word adhesive? Yeah, that's the same concept. Just stick. So more specifically, I'd like to talk about a few examples. So one is exotoxin. The exotoxin are the toxin that's produced by bacteria and secreted outside. So this is cytolytic toxin, and hemolysin is one example. And then you have tetanus and botulinum toxin. And then I will talk about the enterotoxin. Enterotoxin is the toxin produced by enterobacteria. Specifically, it's cholera toxin. So we're going to talk that. Now, in the lab, you should already done this experiment. No, we didn't do it. Did you do it this time? Anyone?
I think so.
Yeah, because I think we were not able to get blood alga plate. And so just so you know, the you can get this is a very classic example. So streptococcus can produce a toxin that can lyse the red blood cell. And so what will happen is that we will put sheep blood, it's not human blood, we'll have sheep blood mixed in agar. It's manufactured by, we use it in unknown labs? Okay.
Yeah, we're using it specifically for the new unknown. We just had to look at pictures for the old one.
Oh, okay, good. So then what we typically do is in the lab, we typically have you do the throat swab, and we let them grow. And then never fails. There's always going to be someone in the lab will have a streptococcus that produces hemolysin, and then lyse the red blood cell, so that you can see this black ring. The black ring is actually a transparent ring. The reason it's black is because this photo is taken on a black background. Okay, so if you hold up the light, this is the area that's transparent. all the blood cell is being lysed. And so this, the one that hemolyzing, so the degrees of effectiveness of the hemolysin, and the beta, alpha, beta is the one that's strong. And this is probably the alpha that has a little bit of lyse, but not all. Okay. So that's the hemolyzing. And so when you look at the unknown, and you'll be able to characterize what what your bacteria is, the unknown, whether it's produced hemolyzing or not. All right. So the next is the neurotoxin I wanted to tell you about. And so before we get into that, so our muscle goes by, you contract and you move your muscle because your muscle, you can tell it to contract and relax. Okay. So when you say contract and your mind says contract, and that gets translated all the way down to the neuron, and then to the interface between muscle cell and the neuron, and that's releasing acetylcholine, and then the muscle cell contracts. And when you say relax, okay, and then the inhibitory neuron actually releases glycine, and then these acetylcholine stops. Okay, so now and you relax, and that's how you go contract, release, all right, relax. However, if you so this is again, it's this is normal, but when you have when the botulinum toxin is present, what happens is that it blocks the release of acetylcholine. So when you have botulinum toxin around, your muscle just keeps relaxing and relax and relax and relax, right? So and this is actually pretty vivid. This is wrinkle. This is wrinkle. This is no wrinkle, right? And so that's, and that's what people use them for, and that's probably where you heard from. And the problem is that if you actually have a bacteria have produced a large amount of toxin and it circulates into your blood, and your heart and start relaxing like this, then you relax too much and your heart stops beeping, and you're dead. So that's why this toxin is it kills people when it has too much. Not antagonist. It's binding to the release of the pore. It's not competition. So, so to answer the question, but so if we were to look at another in the opposite direction, the tetanus toxin. So normally, when you tell your muscle to extend your arm, you're going to have to relax the muscle, right? And so you then have your the glycine gets released at the inhibitory part, and except that the tetanus toxin binds to the inhibitory. Okay, so it binds here. So now your muscle never gets relaxed, and it's always contracting, stuck in one particular state. And so if in the in World War I, they just invented they have to get food to the front line, right? They invented metal can, and so they cook the meat and then put them in a can, and then ship them. The problem is they didn't know that the manufacturing environment needs to be completely sterile because there are bacteria in the air. So quite often, the meat actually, the can will actually have Clostridium bacteria in them. And then what happened is the soldier in the front, because they run out of food, they smell the meat is bad and say, well, okay, let's just cook and get rid of the bad smell, and we can then cover it with seasons and we can still eat. Except in this tetanus toxin is very heat liable, stable. You cannot kill them. You cannot disable it. So it still goes into the soldier's blood, and if they eat food, and then people, well, when they have too much, this toxin will happen. Is that the back muscle will actually fall back, and so strong because the back muscle is very strong, and then they tight, and so you arch people. The patients actually arch backwards and get killed, right? Because your heart couldn't beat because you cannot relax and contract, right? And so that's what gotten the attention. And so now the food is produced always canned in this sterile environment. And so, but it still needs to pay attention when you open a can, especially meat, because these bacteria tend to ferment grow in the meat can. So if it smells bad, you need to really get rid of it, not trying to rescue it. Okay. The next toxin I wanted to talk about is cholera. Cholera has been around for a long time. The first outbreak of cholera actually took place in London. I actually read a book about it, how British people were living in the city. Back then, London has no citywide sewer system, and so their basement is their septic tank, and so they have people come around and collect the sewage, and then and then they also drink. There's no water system either, so they had to drink well water. And so imagine the combination of surface contamination and eventually and get bacteria and then leak into the well. So eventually that did happen. There were an outbreak, and the bacteria gets into the well, and people drink the water and spread, and then they have diarrhea. And then it's everywhere. It's local and then gradually expand. It killed a lot of people. And so what happened is that when bacteria is growing in someone's intestinal tract, and the cholera toxin will be produced by the cholera, right? And then binds to the GM1 receptor. And then and then ultimately what will happen is that the ATP, the adenylate cyclase gets activated. And then the ATP, lots of ATP turns into cyclic AMP. And then and then the result is that they block the sodium from being taken up. So the sodium gets blocked, and then the chloride gets pumped out to match the sodium, and so now in the lumen. So this is in the lumen of the intestinal tract, right? So that's here, and so sorry. So now you have sodium chloride, and when you have sodium chloride in the intestinal tract, high concentration, and we know the water is the only molecule that can go across membrane with ease, right? And so then you have lots and lots of water, and your intestinal tract is not built to hold water, and then you have diarrhea. And and you have people when they have cholera, they actually die of dehydration because they keep losing water. So so the first immediate treatment when they have cholera is to have IV introduce water so that they can stay hydrated. Okay, and so that's the cholera toxin and its effect. Now back to so one can ask the question, why does this cholera causes diarrhea? Is there any benefit? Why would this bacteria have a toxin that causes diarrhea? Anyone to take a guess?
I know that it's gonna be like something that's beneficial to help them like gain more nutrients and reproduce quickly.
How? Because you're coming out of the system, you're coming out of someone's intestinal tract, right? You're going into the nature where there's a less nutrient than in the lumens, right?
Mm-hmm. That's the only part that I was a little confused about, because before we talked about it being in blood. There's no blood here.
No, but so this is where you need to think about microbe. In the past, we always treat microbe as individual, right? And but here you want to think about microbe as a population. As a population, if a population needs to grow, will happen is these cholera will go out of one person, and then it will infect another person, and it will reproduce in another person. So the whole human population gets infected. But this, at the same time, this bacteria population gets expanded, right? So yes, at individual level, they will need to get food, but if they come out, they get a chance of infecting another human being, and when they can reproduce in the other individual, and that's one way of expanding their population as well. So this is actually one way to a good place to remind you that when we think about bacteria, sometimes you need to think, the first you need to think about individual, does it benefit individual bacteria? The next you need to think about, does it benefit the whole species, right? And this is having a diarrhea, well, not immediately after the infection, right? After they reproduce to a certain level, and produce enough toxin, they will cause diarrhea, they will come out, and the remaining bacteria in the lumen will continue to infect and grow and get the nutrient, while the half of the bacteria will come out and trying to infect the another person. Right? So so that's what we want to talk about, the exotoxin. And then you have another class of toxins called endotoxin. Okay, endotoxin. And endotoxin, let me go back to here. Endotoxin. So endotoxin is part of the cell wall. And so normally the endotoxin is made and kept inside the bacteria. And only when the cell die, and then they release. And so in this case, it's the lipid A. It's the lipid A component of LPS, lipopolysaccharide. That is the endotoxin. So if you've heard about sepsis, right? And so where you have a bacteria infection, and if you kill all the bacteria, you'll get a sudden drop of your blood pressure, and that is in part due to the release of the endotoxin. Okay, so so when you have when you treat people when you treat people with a heavy bacterial infection in the blood, and that's one thing that you have to be concerned about. Yes, go ahead.
Can you repeat what you said happens when you kill all the bacteria? Like what happens?
Yeah, when you kill all the bacteria, the endotoxin will be released, like the lipid A will be released, and that's causing a sudden drop of your blood pressure, and that can kill you. And so that needs to be, the doctor usually keep that in mind. So now I wanted to spend time talking about from the human side. We live in such a dangerous world, and what do? So the defense mechanism that we have the general and the specific. So the general can be further divided into innate inflammation and fever, and then phagocytes. And so those are the ones I'll be talking about. Hold on one second. We're going to try to offer an immunology in the lecture in the next semester. And so so if you're interested, you can go and take that class. So I wanted to start discussing the what is innate resistance. Innate resistance referring to these conditions, that the physical barriers and the conditions that prevent other bacteria from colonizing. So so the physical barrier are like the skin, and then the removed particle from the mucus. So I'm coughing now. Is is my physical reaction trying to remove all the the mucus in my throat, right? And that's what you normally cough. And you cough, and the mucus generally goes down from your lung to the to the digestive tract. And so, and normally majority of them don't come out, right? And then the stomach virus is one, and then you have a normal flora, and so that's in the intestinal tract that's competing with bacteria. And so, so when you go to a hospital, when you take on antibiotics, it's actually antibiotics that will suppress the normal flora, and that makes you much more susceptible to other bacteria infection, like C. diff, right? Clostridium infection. That's more associated with hospitalization if you stay in the hospital and on antibiotics for a long time. And then there are other features that we have, lysosome in the tear. Okay, and so it is very important that you have high concentration of lysosome to prevent a bacteria like E. coli or other parts of the body getting to your eye, right? So, so now you understand you shouldn't cry, or if you want to cry, don't cry too much, and you certainly don't want to use your finger to rub your eye, because that's you bringing bacteria to your eye, right? And so these are the features that forms the natural barrier. The inflammation and fever are another way our body trying to kill the invading microbes. Inflammation is always associated with the local infection. So if you have a cut, right, and the bacteria on the surface will get into the blood vessel, and then your body will react. Later on, will show that will have phagocytes trying to kill the bacteria, and so that you have a red inflammation that's localized. And so now you understand that part. Then when you have a cut, what do? When you have a cut, you actually want to have let the blood run a little bit. Don't get so you have lots of blood. You have 5 liters of it, and so you just let the blood drip a little bit, and so then your blood pressure actually will wash the wound or the cut actually better, and let it drip, and then you stop the bleeding. Okay, don't. Now obviously if you have a giant break, you have lots of water, blood coming out, then you'll want to stop, right? But typically you'll have a skin paper cut, and and then you will let it run a little bit, and so the flood of blood actually will ensure the washout or increase the chances the bacteria gets washed out. Now, obviously, there are other conditions will make it difficult for platelet to work and take longer to heal, and those are the conditions that you want to be very, very careful. So that's related to inflammation, and fever is a systemic, the whole body reaction, and your body, once it detects the bacteria, debris, or bacteria in the blood, and your body, the entire body, react to raise the temperature so that you can have your enzyme and your other cell to work over on a more efficient way to kill all the microbes. So fever is like your systemic reaction or last defense, right? If you read stories of early humans, they were going to high fever, and because back then there's no medicine, right? And you can't have antibiotics. But now that's why now it's not important to have the fever, and you just, when you have fever, just suppress it. However, you do want to make sure that you understand when you have a fever, that's an indication that you have infection somewhere, and so that it's important to seek treatment. Okay. Now, the next, the general response I wanted to talk about is the phagocytes. The phagocytes are the cells that goes around and capture bacteria and kills them. Okay. And so to understand those, then I'll bring up this slide. And among the slides that I'll be presenting in today's lecture, and this one slide is about one of the most important slides, right? Now, sometimes I say, well, this slide is so important that you need to be able to get a piece of white paper and be able to draw this slide. And this is one of those slides that you should be able to draw. What do you draw? Well, you need to understand the cell types. So you have bone marrow, start with bone marrow, and then it goes to myeloid precursor, and this goes into the blood. And then you have monocytes. Then the monocytes can differentiate into macrophage. So for example, the monocytes are circulating in the blood. And when you have a cut, your blood vessel dilates, and these monocytes will then leave the blood vessel and come out and to become a macrophage. And the macrophage has a rough surface. And so for example, you can imagine a bacteria can fit in here, right? Right? And so this is a macrophage. And then you have other cell types, neutrophil, masters. But for us, this part is very important. And later on, you will, when we talk about the cell-mediated response, and this part becomes very important. Okay, you must know the cells goes into the lymphoids, and then mature into T cell and B cell. And the B cell, once it's activated, selected, and then goes into plasma cell. And the plasma cell is the one that produces the antibody IgG, and that's everyone's talking about. All right. So to go to, so that's the cell type. Now the macrophage is actually functioning in both the general defense mechanism and the specific. And so the macrophage is also considered as the antigen presenting cell. Okay, antigen presenting cell, APC. So here is an example of the red blood cell with some, this is monocytes and different cell types. And so here is how does macrophage work? And so the macrophage takes up bacteria. So when bacteria gets taken up, and they are in the phagolysosome, and the and then the bacteria, the phagolysosome is exposed to hydrogen peroxide and all the enzymes and reactive oxygen species, and that's what kills them, right? And this is why when you get a cut, you use hydrogen peroxide, these in the brown bottle, to clean the wound, right? And so you can actually see what's happening. Here's an example of bacillus gets engulfed. This is a chain of bacillus bacteria. And you're starting off here and taking up the bacteria, taking up more, going inside, but you focus on here. You can see the white area. And the more you go in, if you compare to what's inside versus outside, and eventually these bacteria gets lysed. They've taken up the entire bacteria and they lyse. Okay, so this is the bacteria. The macrophage, what does it do to take up? Now, later on, we will be talking about macrophage can be activated. So this is at a smooth surface or have a little little fold to take up the bacteria. So it's like, if you look at my hand, can you all see my hand? Right? And so if you, later on, you will talk about, normally the macrophage is like this, okay? You take up the bacteria. But later on, we'll talk about that macrophage can be activated. And then once it's activated, you have multiple folds. And then you can take up additional bacteria. Can you see? I'm taking up more bacteria in my hands. All right. So, okay, good. And so this is I'm using this graphics as an opportunity. Okay. So the next I wanted to introduce the T cell. So remember, there are two main types, the T cell and the B cell. So we're going to talk about the T cell first. So, and then we're going to talk about the cells, how to interact. So the T cell, there are two types of T cell, the T cytotoxic cell and the T helper. And the T cytotoxic, by definition, is producing toxin to kill, and the T helper is the one that recognize and act. Okay, so one way to think about this is one T cell is to help, one T cell is to activate. Well, obviously the one cytotoxic one will be the kill. All right. Now, there are surface receptors. They have T cell surface receptors. And the T cell surface receptors are different. One is CD8, one is CD4. And then the T helper there are two types. They specialized. One is to activate macrophage. One is to activate the B cell. Activating macrophage will making the macrophage more effective in capture bacteria. Activating B cell is to make the B cell differentiate into plasma cell and duplicate and make it more. So that's almost that's what the vaccine is related to the vaccine. Ania, yes, go ahead.
Can you repeat what you said about the B cells one more time?
About B cell?
Yeah, when you said it activates the B cells and something else. I'm trying to write it down.
Right. So B cell will come down to talk about the B cell. So activate the B cell, and then the B cell will replicate to make additional B cell and then make plasma cell. Okay. And so these are the activation part. So now comes into the hard part that you do actually have to understand is that the T cell doesn't work alone, does not work alone, right? So the T cell is to kill. Well, the kill, you can't just kill any cells, right? You need to know which cell to kill. And to help or to activate, you also have to know which cell to activate, right? So now comes the part that you absolutely need to concentrate and you understand. So here is on the top is the T cytotoxic cell. Okay, and then here is the target cell that to be killed. So this is how you kill, how T cell kill. And then on the T cell receptor, there's a T cell receptor on the surface. And the T cell receptor has to have an antigen. And then you have the Mhc complex. It's called major histone compatibility complex. It's a mouthful. That's the receptor on the cells, on the cell that to be killed. Every cell has this Mhc complex. What's important here is whether or not the cell has the antigen. Okay, so in order for this interactions to stabilize, you also have CD8. So you have T cell receptor and CD8 for cytotoxic. And so now when you have stable interaction, then the T cell produce granule, and this is the trigger to cell program death. It's apoptosis. They don't actually kill it. They trigger the cell to kill themselves. Okay. Now, as I said, there are lots of cells in your body, and which one should the T cell grab and kill? And that the critical part is the antigen. So let's take a look. And so this is the antigen to be recognized by the T cell receptor. The T cell receptor has two parts, the variable region and the constant region, okay, in blue. And so these are pre-made, and so that the T cells, we have lots of T cells. All the T cells have the same constant region, but we have different T cells with different variable region, and they can match different shape and size of the antigen. On the other side is the major histone compatibility complex, the Mhc complex. All cells, all the nucleated cells, all cells have this class 1, but the class 2 is the only one that have on the surface of macrophage and B cell. Remember, these are the ones that can be activated by T helper, and the T helper recognize this class 2. So all the cells have this, including the B cell and the macrophage. So macrophage can also be killed, the B cell can also be killed, but the rest of the cell, if they present an antigen, they will be killed. So so this is how it works. So when bacteria kills the captured an antigen and then kills it, what they will do is then they will take parts of the protein and then put it on the surface. So this is I'm sorry, this is internal foreign proteins. If all cells capture internal protein, they will present here, and then the T cytotoxic cell will identify it and then kill it. So to kill this cell, all right. So now the question is, why would we do that? So this is a double security. So if a cell have internal fluorine protein, because they take up the cell, they take up a protein, and then they can take up a protein or foreign invading cell or virus, and then we then tell that particular cell to go self-lyse, so completely wipe out any possible contamination. Okay, so that's the reason you want the cell that has the foreign protein to be killed, right? Now, how do you know? Well, the only cells that have the antigen, right? Because you have to have an antigen. The cells that has the antigen are the cells that have taken up foreign object or foreign protein. Okay, so now this is kill, right? So but then you have the macrophage. All right. So the macrophage, in addition to be killed, they also can be activated, and that relies on macrophage has the class 2 Mhc complex. So the class 2 works by this. They will take up the foreign bacteria and they will digest, and then they will present the antigen at the on the class 2 Mhc complex, right? And so that class 2 is then recognized by the T helper. Remember, the T helper has a different T cell receptor so that the T helper will get here. All right, and so then the T helper can bind and then activate the macrophage, and this activation allows the macrophage to be much more effective in taking up. Now, once they take up more, as the macrophage taking up more foreign bacteria, and because they also have class 1, right? And they can also be recognized by T cytotoxic, and then these cells will be eliminated, the macrophage, right? So now you have a system. So we actually have a system that the macrophage goes around, capture the bacteria, lyse them, and then go around, tell T cells, say, hey, I just killed a bacteria, and so I am contaminated. Please get me eliminated. So I no longer pose any sort of threat to the body, right? So so we have this double security. So now, what about the B cell activation? Well, okay. So if you look at the B cell actually have antibodies and the T cell receptor, the Mhc complex too, right? And so what will happen is that they will present the antigen on both the Mhc complex here. All right, they will present the antigen here, and then and those will be activated, and those B cells then will be activated, and to make more memory cell, and then the plasma cell. And the plasma cell is the one that will produce lots of IgG. And on the surface of B cell, and these B cell will produce IgM. Okay, that's why when you were testing the initial testing for the COVID-19, they test IgG and IgM. If you're just being exposed once, then you only have majority is IgM. And if you get multiple exposure, then you have IgG. Okay, so that's the T helper and T cytotoxic cell. So now I wanted to turn to B cell, because we just mentioned the B cell and produces different antibody. So you will often see this particular picture or diagram, and this IgG, immunoglobulin G. And so IgG is the most important antibody. Okay, you absolutely need to know how this works. Look at this picture. You need to dream, be able to draw this picture. And if you draw IgG incorrectly, you basically tell people that you didn't learn, you didn't know about immunology. All right. So IgG has 4 chains, 2 long chains and 2 short chains, and they are linked together by peptide bonds. And they have, each chain has constant region and variable region. When it's constant, that's constant among all human cells. So you and I will all have the same constant region. All humans have the same constant region. But individually, even within your cells, you will have lots of different B cell. Each B cell will have one set of the variable region. Okay, so it is important that you understand where the variation is. The variation is at individual B cell. So when one B cell recognizes COVID-19, and that one gets activated and reproduced, and so that's where the clonal selection concept comes in. Clonal selection. And so the B cell then produces plasma cell, and the plasma cell produces lots of IgG. And the scale of this antibody is also important. And so this is a protein, and the part that recognized by an antibody variable region of the antibody is called the epitope. Okay, epitope are the ones that it's typically somewhere between 3 and 6 amino acids. And so that gives you a sense of scale. A typical bacteria protein is about 300 amino acids, right? And only 3 to 6 is being recognized by one antibody. Okay, so you can have lots of a protein can have lots of antibody recognize it. And so when you inject a protein into an animal or us, you're going to elicit polyclonal antibody. Like lots of B cell will recognize and then produce lots of antibody. So you can have antibody recognize different parts of the protein. Okay, so that's why it's important. Now, the but you can have monoclonal antibody, right? But that's a bit risky. So what's monoclonal? Monoclonal is just one antibody. Okay, so Regeneron produces a one monoclonal antibody recognizes COVID-19, the S spike protein. Imagine this is the spike protein. Then you can have, if you inject the protein into your body, then you actually have lots of antibodies being produced, right? So your COVID-19 vaccine injects mRNA, so you actually produces this spike protein, circulates in the blood, and select lots of antibodies, B cells to be activated, and you have polyclonal antibodies against the COVID-19. And Regeneron then produces, they do the selection and they pick one particular strong antibody. They will test which one is most effective in blocking the COVID-19, right? And then they use that as they make a huge amount, manufacture a huge amount, and then they inject into your body. That's how they work. And in contrast to IgG, IgM can be linked to, 5 of them can be linked together to form IgM. The difference between IgG and IgM is IgM is actually the ones on the B cell surface. And IgG is the one that's produced by plasma cell. And so if you look at the reaction, what happened is that when you first inject or first exposure to the antigen, you will have the IgM, mostly IgM, it's being produced. And then it's the second exposure that allows you have B cells to produce lots of plasma cells. Lots of plasma cell immediately produce lots of IgG. And we know over time IgG will reduce. However, in theory, the plasma cell, not the plasma cell, the memory cell will be there. Lots of B cells will be there. And so when you have the second exposure, then you can quickly produce huge amount of the plasma cell. So the difference is that imagine normally you will have one B cell recognize COVID-19, right? And when it comes in, you naturally, if you exposed to COVID-19 and that one B cell gets activated, selected, and then you produce IgM, and then produce 100 B cells that recognize COVID-19. Right, that's one vaccine. And so, but when you have a real vaccine, a real COVID-19 comes in, instead of having one B cell, now you have 100 B cell that recognize the COVID-19, and then your body reacts very fast in producing lots of IgG, and then covers the virus with the IgG and prevents it from infecting further, right? And so normally that's how it works. The IgG and IgM works. Now, in addition to what you already know about the function of IgG, I wanted to expand one more, is what's called the complement system. How does antibody contributing to the killing of bacteria? So I want to focus your attention on the top. The classical complementation activation. The classical. So what happens is that if you have 2 antibodies, here is the part that you need 1 and 2. You need 2 antibodies on the surface of bacteria, side by side, not far apart, side by side. Okay, and then that allows the C1 complement 1 protein to come and attach to the antibody. Okay, remember this is the constant region that is being function as the anchor. So the C1 then recruits C 2, 3, 4, 5, and then they all come. And then the net result is that the C5 and C5 and C9 protein 5, 6, 7, 8, 9, they actually form a ring and punch a hole through the bacteria cell wall and cell membrane. Now, and then the bacteria can have multiple holes, right? So if you have a bacteria have lots of holes on their surface, is the bacteria still alive?
No.
No, right? Because all the things leaks out, the enzymes leaks out, and ATP leaks out, and all the good stuff leaks out, right? And so then the bacteria is dead. Okay. So these are the system. So we for the specific host defense mechanism, we talked about it's targeting using T cells and B cells to recognize specific cell types to function, to act, right? Involve that. And so that's the specific. So now I wanted to talk the whole immune system as a whole. How does it actually work as a whole system? Okay. So yes, we talked about individual components, but they work together, right? So here is a quick overview of how this system works. So if you have the cell and then exposed to antigen, so you have proteins or bacteria or virus, and they all captured by this APC and the antigen presenting cell, which we also know as the macrophage, or any cell that can actually capture the bacteria or foreign protein, right? And then what's important is that they kill these bacteria and then they present the antigen to the surface. Right? There are two types, the MHC1 and MHC2. And depends on which one is being produced, and they are then recognized by different T cells. Okay? There are two types of T cell, cytotoxic and T helper. And then you sometimes wonder, well, what about a macrophage? What does it do? Does it get killed or does it get activated? It's a statistics. It's a frequency thing because in the blood, these cells and everything is floating, right, in your blood, and so the chances of them bumping into each other determines whether a macrophage gets killed or gets activated. Okay, so some get activated, some get killed immediately. Some, even the activated one, will eventually bump into a T cytotoxic cell and gets killed, right? So here you have T cytotoxic cell. If it gets, then the T cell will go and activate. If it's an activation, or this is if it's an activation, then what happens is the B cell gets activated and produces antibody. And then the antibody will bind to antigen. Now, if this antigen is on the surface of a virus, then this antibody will basically cover the whole virus with antibody, right? And then the spike protein no longer binds to the receptor, because now you have it covered, right? And so then the binding, you can have now these antibody depends on where the binding is. If this antibody is on the surface of bacteria, then you can trigger the complement, right? And that kills. Now that's just one way of kill, but you also can actually help the phagocytosis, help these macrophage, because they go a process called the opsonization. Opsonization is a process that have protein antibody attached to the surface of bacteria, and then the bacteria becomes rough. The surface becomes rough, and then they can be easily picked up. Just like the way you think about this is a new soap, which has logos imprinted on them. If they're in the water, it's easier to pick them up, right? Because it's a rough surface. But if it's a used soap and just it is very slippery, right? So imagine if it's a slippery soap, and if you stick some toothpicks to them, and that will make it very easy for you to pick up, right? So that's the same concept of opsonization. So opsonization is the antibody makes the surface bacteria rough, and so then the bacteria then can be taken up much more easily by the macrophage and then gets killed. So on this side, and that the T cells can activate T cells, and the T cell then goes to trigger other cells, goes into inflammation, and then you can have cytotoxic killing, and then the antigen gets destroyed, and so that goes into that cycle. So either it kills directly or activates, and these system works together. And so that brings us to the final slide. This is I want to end the class, but I wanted to highlight that yes, we talk about these things individually, but they work together. Different system, the general host and the specific, and the general involves in recognizing and then kills, and then these inane the system, you have even harmful bacteria, these non-specific that they can be blocked. So all together, you see the your microbes, when they get to human, they first gets blocked or gets all competed, and if they not get all competed, they are then get captured by the macrophage, and then the macrophage kills, and then triggers the activation of specific host defense mechanism, and then kills these bacteria. So altogether, we actually have a very strong immune response and defense mechanism. So that's keeping us healthy, even though the bacteria is on our surface. And so hopefully with this lecture, you can sleep much better and say, aha, I have a very strong defense mechanism. I am not afraid of microbes. All right. So I like to end the lecture here.